Hello, everyone. This talk is part of a series of minute talks by the member of the consortium Evopas Amazonia. And just to recall you that you can find all minute call of this consortium in their website on the YouTube button. My name is Christine Chevillon. I'm a CNRS researcher working in southern France in a structure called Mycobacteria, which, as usually in France, merge some academic staff from different institutions, namely the local university, Montpellier University, and two national institutes, the CNRS and the IRD. I was initially trained as population geneticist and starting my career on the thematic of the genetics of adaptation. And along the 15 past years, I've been interested in understanding how the articulation between parasitic adaptation and parasitic emergence. This gave me the opportunity to work with different researchers with very different backgrounds than I, such as virologists, for instance, but also entomologists, veterinarians. And this is really what I like, because I think that it's a dialogue between the different dominant scientific fields that really make science progressing. And this is actually the main strain, at least to my eyes, on the consortium Evopas Amazonia. We are not only academics and students, postdocs from different French and American institutes, but the most important is that we have complementary skills in diverse scientific fields such as microbiology, ecology, community ecology, macro and microevolution, but also genomics, for instance. And this gave we try to integrate all these skills to develop the project Novel Weapon Evolution in Mycobacterial Pathogenesis, Dispersal, and Ecological Persistence. When we say Mycobacteria, usually it's more a background of medical science, veterinary science that consist in the largest literature. So as previous minute talks were giving some broad picture of the different set of questions addressed by this consortium, the aim of the today's talk is more to explain, to see the version of what is a Mycobacteria with the lens of microevolutionists. So when we are tracking what are the forces that make their population to evolve. Mycobacterium species are very diverse. They have recurrently adapted to diverse micro-niches in aquatic and soil habitats. And so in fact, they are really ubiquitous in aquatic and soil microbial communities. The lifestyle, in fact, can be pictured by these two pictures. The first means that they are regularly cycling between planktonic cells, they aggregate, attach to something, and form biofilm, where either alone or with different other microbial species that maturate and hatch and disperse and going back to planktonic cells. Another common trait of Mycobacteria is that they know how to resist predation by some microbial predator called amoeba. And this even in the cyst stage, so in French is cyst, which is a form of amoeba able to to be dormant when the external conditions are too stressful. And so for the Mycobacteria, they can, by resisting to this predation and maintaining alive within the life of the cell of the predator, they can really be protected against harmful environmental conditions, and they would germinate later on and go back to form biofilm and so on. In fact, we know the genetic basis of this innovation, to resist predation. It's a type 7 excretion system that is specific to Mycobacteria. In fact, it's a family of genes, and it's a copy ASX1 that allows intracellular life. And this opened also a new road, because when there are spillover into vertebrate, whether it's bird, mammals, fish, they can also enter within vertebrate cells and live there. And in fact, this ASX genes family, the copy 3 and 5 are also involved in pathogenesis through the formation of granulosome or polymorphism called rough and versus moss. And from the spillover into a vertebrate can have two solutions. True host are those that are allowing the return of the bacteria to the global environmental population. In that case, the vertebrate is really a host or a reservoir, if you prefer. It doesn't what the focus you want to put in. But if there is no return possible, and if the bacteria is trapped within the vertebrate, in that sense, in fact, the vertebrate is said to be a dead-end host, and so the bacteria is just a biological pollutant. In fact, the evolution of the Mycobacteria population is totally indifferent to what happens in the spillover of the vertebrates. In Mycobacteria, there are, in fact, what is interesting in literature, if you look at it, in fact, is that the increase in spillover frequency correspond, in fact, to perturbated and unstable microbial community, usually colonizing some artificial setting, and so for very diverse Mycobacteria. There are two exceptions in this global picture of the life of Mycobacteria. Two independent events that is really an evolution toward a true specific lifestyle. The first event occurred at the beginning of the lepre complex, and in fact, these members have lost the ability to live outside cells. So there is no biofilm anymore. They can have an environmental reservoir, especially within the cell of microbial predators, but most often they are really cycling from a human to another by nasal secretion. And in some cases, in some areas, they can also circulate from an animal to another. For instance, in the New World, the night bands, Amaldio is a good animal reservoir. So far, we don't know really the frequency among this, the frequency of transition among these three compartments. The second evolution toward a true parasitic lifestyle was seen for the members of the tuberculosis complex. And in fact, the ancestral seems to be nearby, very similar to the tuberculosis sensu stricto, where the contamination goes from one human to another. Here too, there can be an environmental reservoir with we don't know very much of the importance of it. And for the other members of this complex, usually they have loose parts of germ, so this seems more evolved, but second evolution toward different host specialization, circulating, for instance, in sheep or in cattle. But this host specialization is not very strict, so you can have some passage from the normal host to another. Another type of exception is formed by the mycolactin-producing Mycobacteria that we are studying in this consortium. And this MPM is part of the marinum complex. The marinum sensu stricto is live exactly as the other Mycobacteria. So between infected fish, forming biofilm, resisting to predation within amoeba. But a few genotypes of this marinum complex have lost the capacity for intracellular life because they have lost the ES1 gene. And this one, they have gained a plasmid that is coding the mycolactin toxin, and these are the MPM producing Mycobacteria. They have given different names depending on the geography of isolation and on which host they were, on which vertebrate they have been isolated. All these MPM are also showing two different traits, which is a genomic rearrangement, extensive. Even here, you can see the pangenome tree, and you can see that the rearrangement goes on within the MPM, and that in fact, they are very close together from one to another. Looking at the life cycle, in fact, for MPM, human and other mammals are dead-end hosts, so they cannot be a reservoir. They are just a non-U-turn road where it can be loosed and can suffer from infection, but do not participate to the evolution of these Mycobacteria anymore. The plasmid of virulence beared by this MPM, we do know that it is associated to a high fitness cost, making the MPM very poor microbial competitors within the microbial communities. But in fact, when ecologists and our scientists just look at the presence in some habitats, this species, which is a poor competitor, can really dominate and see associated in very different species within aquatic communities. Different macroecologists, including some who are part of this consortium, as Eric Bembo or Jean-François Guégan, have started to really try to understand what make these communities invaded by Mycobacterium ulcerans characteristics. In fact, they have seen that it's not always the poorest communities in numbers of species or species diversity. They also showed that, in fact, this invasion can be fully random, and depending on some key abiotic factors, they can switch from random transmission to any species in the community toward another way, which is a trophic transmission. Complementarily, in the lab, where some experiences showed that this plasmid can be lost or can be unexpressed, and that the presence of chitin in microhabitat can change the competing balance between this Mycobacteria and all other microbial competitors. But this is just a start. What we will do in this consortium is really to try to integrate all the different events on the changes in the structure of microbial communities, but also the communities of macroorganisms, to see what is the series of events linking human-made perturbation of the ecosystem to the shift that make a poor competitor to a dominating biological pollutant. Okay, thank you for listening. Bye.